Hi, everybody. So, welcome. Thursday, January 21st. It is our last class of the first semester. Yay! So, what we're going to end up doing today are a couple of things. So, we're going to take a protein synthesis quiz that will kind of go on first semester. So, we're going to do that first in Schoology. It's 3 total questions long, worth 8 points. And it's going to go along mostly with your entry number 13, maybe a little bit in entry number 12. So just kind of have those in front of you if you need to reference them. And then we're going to finish out Walter, which is entry number 14. And as I mentioned, it is the last day of the semester, so between now and hopefully end of school on Friday, you will kind of share, submit, upload any and everything that you have that's either missing or partially completed. Okay, so we'll do the quiz first in Schoology. If you are not present in class, that will be open to you from class time onward. So you can kind of go in and take that at any point in time, and then it should let me know when you've taken it so I can see the score. Otherwise, I'm just going to bounce into our presentation of Walter, if it lets me here, and scroll to day 2. So we didn't get very far last time. We talked about his symptoms and what was wrong with him, but I'm going to hop to day 2. So I want you to take a minute. This is really where we left off last time, and see if you've got your A page set up in a similar fashion as this. So if you don't, go ahead and hit pause and set it up, and then play to continue. Okay, so if you are in class or not, we are going to take a break here and really discuss possible diagnosis. So I'm going to just kind of open up the chat in class and have people shout out or type based on the symptoms that we learned on Tuesday what they think is wrong with Walter. If we're going to put a condition or disease on it, what would it be? So we're going to spend a couple minutes just talking about possibilities, and then we're going to look at the blood work. So the test results, and that is going to be on the next slide. So here's what we get. And I kind of have 2 fields of view. So in this particular blood work, what they are doing is they take basically a drop of blood and then they put it on a slide and put it underneath the microscope. And this is what they see. So on the left, we have a healthy comparison, meaning like a healthy individual or a blood sample from a, healthy human. And then on the right, we have Walter's sample to compare. And so I'm going to ask you now to kind of pause and draw those into the circles on your 14A notes page. And you don't have to draw like every single cell, but just kind of a representative picture. You can kind of see later on in my own notes picture. I will paste in later. I don't have it in there right this second. But you'll be able to kind of see how thoroughly I did it. So just kind of go through and draw what you see. And as you do so, ask yourself, like, what observations can you make? Based on this, is Walter looking great? Should we be looking elsewhere besides his blood, or is this seeming like the issue? So as you're drawing the picture, think about that. And then pause if you need to. And then when you're ready to move on, go ahead and push play. Okay. So now that you have the pictures down and we have Walter's sample here, I want you to remember, we've learned structure equals function. And hopefully you've been able to really pinpoint here that, yes, there does seem to be a problem, right? Like the cells that I'm circling right now are not those beautiful circle red blood cell shapes that we're used to seeing. We have these weird kind of elongated, here's one down here that's particularly hook looking. Something's going on. So it appears as if some of Walter's red blood cells are not shaped properly. And so if Walter's blood cells aren't shaped properly, then it probably makes sense that they're not working properly. And I want you to stop for a moment and think about, what do red blood cells do for us? Would this fit with Walter's symptoms? We know that he is short of breath. We know that he has pain in his joints and his ankles, or joints and muscles, my mistake. We know that he was kind of a sickly kid. He's got a yellow tint to his eyes. Would poor functioning red blood cells in any way go along with those symptoms? Let's learn a little bit more about red blood cells to see if we can make a better guess. So in this particular picture, I have a red blood cell, and you can kind of see it's got a characteristic, like, I don't know, lifesaver shape. It's like a circle with a little kind of dent in the middle. And when you zoom in really close, you can see that red blood cells are the oxygen-containing component to our blood. And you can see here the main protein that is found in red blood cells is hemoglobin. And hemoglobin is that magical protein that is able to trap and hold and carry oxygen throughout the body. So it's the super awesome thing that is inside red blood cells and carry oxygen to all the parts of the body that we need. Sorry, I'm choking on a piece of food in the background. I promise I'm not always like this. And also then drops off oxygen to keep us going. So again, I'm going to ask you, structure, function, if these red blood cells aren't shaped correctly, or if proteins in these red blood cells are not shaped correctly, then they're probably not working correctly. So does the ability to deliver oxygen in various parts of the body, would that have anything to do with the symptoms that he's experiencing? Keep thinking about that. And also think about what might have happened to change Walter's blood cells. How did they get messed up in the first place? What might have happened to get this weird structure in the first place? If I'm saying that the structure is somehow messed up and probably the function, well, how did the structure get messed up? Let's look a little deeper. So I've kind of put Walter's cells on the left and the normal cells on the right. It appears that the hemoglobin protein is not folded and or structured and functioning correctly. It is abnormal in some way. Whereas in normal cells, it has a different particular structure, carries oxygen, all as well. And so because of the abnormal hemoglobin, we get these more moon-shaped blood cells compared to the nice, awesome little circular ones. And so if we think about the fact that red blood cells usually run through our arteries and veins, right? And then even capillaries, which are only one blood cell wide, tight squeeze, how going to these branching locations might be a little bit more tricky for the ones that have jagged edges compared to the ones that are nice and circular and bounce around. You can see they're kind of blocking flow and jamming up. Does this fit with Walter's symptoms? Could this maybe explain why he's got pain in his joints and his muscles? If that's where blood is flowing and it's getting blocked, could that explain it? If we're not able to deliver oxygen where it needs to be, could that maybe explain why he is short of breath or feels soreness? Things might be kind of lining up. So if one of the proteins in Walter's red blood cells isn't folded correctly, I'm going to put isn't because I have a typo there, then it cannot do its job. So if it's not folded correctly, then it can't do its job. And so in the bottom part of your notes page, I want to kind of get everybody to have a uniform analysis. A problem appears to be with the hemoglobin protein. And that seems to be Walter's issue. So based on what we protein in RBCs appears to be the problem with Walter. So I want everybody to kind of stop for a moment, pause, and add that to the analysis because it seems to be that that protein is the issue. And as you write that, I want you to think again, why is the hemoglobin misshapen? What could cause that in the first place? All right, so you should have the analysis down. If you don't, give it a pause. So let's talk about what causes a misshapen protein. If we think back to the central dogma that we have learned of biology, right? We have DNA that gets turned into RNA by the process of transcription. And that RNA message gets turned into the language of amino acids by the process of translation. And then those amino acids fold themselves up and become a protein. So this is how it's supposed to go, right? So how do we end up with an issue at the end of the line, at the protein level? How do we get a problem here? Well, to figure that out, we're going to have to work backwards. Where should we go? Maybe we should go to the source. So let's take a look at Walter's DNA and see if we can find any problems there. If there's an issue with the blueprint, then maybe it makes sense that the protein that results from reading the blueprint is the reason why. So to attack this problem, on your B page, at the very top of your B page, you are going to work through the process of protein synthesis, transcription, and translation. Just like we did in entry 13, exactly the same thing, except now we have a healthy patient and we have Walter to see if this is the source of the problem. So I've given the codes typed out here on the slide. I want you to pause here and jot these into your B page and then leave space so that you can transcribe and translate. And I put the codon chart there for you. So once you get the mRNA message, you can use it to decode the amino acids. Again, this is exactly like we did on 13B. I'm just giving you a different sequence. So as you work through, I want you to see if you can spot the difference between Walter and the healthy patient. And I'm going to work through this in class and then have this up. And I'll post a picture after we go through in class so you can see what the messages should have been in case you are struggling with that part. But the key part is that there is a problem. And what I'm going to end up doing real quick now that I'm talking this out, I'm going to actually delete this part because I want to highlight the key difference. So in the process, you should have arrived at that one spot between the normal person and Walter, where it was CTT in the normal DNA, and then it was CAT in the hemoglobin, in Walter's DNA. There's a change. And that change then moves its way down the line. The mRNA on a healthy person would then be GAA, but for Walter, it's GUA, which means that when we look at our little codon chart wheel, we get glutamic acid for normal people, meaning that the protein will fold correctly. And in Walter's case, the GUA actually codes for a different amino acid. It codes for valine. And so that means or results in the folding incorrectly of the protein. So that appears to be the source of the problem. By working backwards, we were able to identify this one letter change was enough to cause this whole big old problem. So what does that translate to? That mutant or altered shape of the cells, the red blood cells, is called sickle cell anemia. And so that is our doctor diagnosis for Walter. He has sickle cell anemia. And I'm going to kind of amend this a little bit. So he has sickle cell anemia from a mutation, which is a single letter change in this case, in the hemoglobin's gene or the DNA. And that resulted in an amino acid change, right? Glutamic acid turned into valine. And then that leads to different amino acids are then going to fold a little bit differently. And so that's how we get the incorrect folding versus the correct folding. Okay. And so to kind of put it into picture a little bit too, this is another representation where you have the DNA sequence and then you have the amino acids at the end. It kind of cuts out that middle mRNA. But it brings about important things. Unit 3, vocab page. It brings about 2 new key vocabulary words that I want to make sure that you have. One of them is genotype, and the other is phenotype. And we're going to be using these words a lot as we move forward, so I want to make sure that you have them on your vocab page, or you can even put them on your A page here if you'd rather. But genotype is the genetic code. It is the letters that you have for a particular trait. Whereas your phenotype is the physical appearance of that trait. It's what we can see. So in this case, your genotype would be the genetic code for a trait. Whereas your phenotype is the physical appearance, what we can see. So I can't like look at a person and say, hey, they have a mutation. At exactly this point, exactly this letter. But I can see under a microscope their blood cells. Or you can't see a person's genetic code that's, you know, spelling out their hair color or eye color, but you can see their hair color and eye color. So that's genotype, think genetic, phenotype, think physical. So I want to make sure that we get those words in. Okay. So I mentioned the idea of mutations, and so I just wanted to let you know there's a bunch of different ways that can happen. You can, looking at this original message, we can swap out a letter. So instead of time to dream, we have tame to dream, which is what actually happened in Walter's case, a single letter change. Sometimes things get reversed, and then we would have emit to dream instead of time to dream. Sometimes letters accidentally get deleted. That usually messes everything up, right? Because it shifts everything down the line. And sometimes we might add in an extra letter, which again kind of shifts everything down the line. So those have huge consequences. Single letter changes may or may not. It kind of depends on what that new letter would code for. In our particular instance, it changed the amino acid, and the amino acid that it changed for is quite different. And so because of that, we end up with sickle cells instead of normal red blood cells. And then in class, I'm just going to show this video that's like a quick 1 or 2 minute talking about sickle cell. Okay. So the final thing that I'm going to introduce is an article about different blood disorders. Sickle cell anemia is one of them, but there are a number of others as well. So I'm just going to have you read this if you want it bigger. Again, this is just a super tiny snapshot. I have it on a Google Doc. You can just click here and go to it in bigger font. And then I want you to write down 3 takeaways. So just reading through the article, I want you to write down 3 key takeaways of what you learned. And that will totally finish out your B page. I'm going to actually move this up. There we go, just like that. Okay. And so just kind of wrapping up, your goal will be by next week, the start of the semester, to just finish out entry 14 related to Walter. All right, that's it. Thank you.